Well, good afternoon, and thank you for joining us. My name is Mary Lee Watts, and I am the Director of Federal Affairs at the American Society for Microbiology, and I will be your moderator today. We have an excellent lineup of speakers for you this afternoon, and we're going to be focusing on progress towards the development of therapeutic options to treat COVID-19. But first, it is my pleasure to introduce Dr. Robin Patel, President of the American Society for Microbiology, who will be giving opening remarks.
Thank you for joining us today for COVID-19 testing, vaccines, and therapeutics. We have two world-class scientists presenting to you today who I will introduce later. The American Society for Microbiology, or ASM, is committed to bringing you the best science available so you can better understand the current pandemic and make evidence-based policy decisions. We can also provide insight into broader issues within the microbial sciences. Members of the American Society for Microbiology have been on the front lines in the fight against COVID-19. Back in March, ASM convened a virtual summit of international experts in the microbial sciences to set research priorities on diagnostics, vaccines, and therapies for COVID-19. Our speakers today were active participants in that summit. There's great excitement around the development of promising therapies for COVID-19, and today you will learn more about how scientists target viruses both inside and outside the cells they infect, as well as hear real-time updates on progress towards therapeutic options for the disease. As you will hear from our speakers, today's work on COVID-19 treatment builds on decades of basic research into pathogens, how the immune system responds to infection, and how viruses attack cells. Thanks to increased federal investments in basic, translational, and clinical research through the National Institutes of Health and other federal agencies, we're moving at record speed to develop novel and test existing antiviral drugs and immunotherapies. In particular, ASM acknowledges the emergency multi-year funding Congress has appropriated in recent weeks to the National Institute of Allergy and Infectious Diseases and the Biomedical Advanced Research and Development Authority to accelerate therapeutic development. Without this support, scientists like Dr. Dennison and Dr. Casadevall would not be able to advance this important work at such a rapid pace. Before I introduce our speakers, though, it's my pleasure to introduce a special guest for opening remarks. I'm honored to introduce a special guest this afternoon, Senator Bob Casey of Pennsylvania. As a member of the Senate Help Committee, he's consistently demonstrated unwavering commitment to advancing science, protecting children and families, and improving public health and security through medical research and through support for agencies like the CDC, BARDA, and the FDA. Senator Casey also led the effort with Senator Richard Burr to successfully reauthorize the Pandemic and All Hazards Preparedness and Innovation Act. This bill has been absolutely critical to our ongoing efforts to address the COVID-19 pandemic and resulting public health emergencies. But perhaps even more salient to today's discussion, Senator Casey is going beyond his duties as a public servant and doing his part to advance research as a citizen. Recently, Senator Casey tested positive for antibodies to the SARS-CoV-2 virus, suggesting that a flu-like illness he experienced early this spring was in fact COVID-19. Given this development and the research you will hear about in a moment, Senator Casey decided to donate his own plasma to clinical research in hopes that he might be able to help others recover from this devastating illness. The American Society for Microbiology is deeply grateful to Senator Casey for his personal and professional leadership on many levels. Thank you for taking the time to share remarks with us today.
Dr. Patel, thank you and the American Society for Microbiology for this opportunity for me to be part of this briefing, and to thank you for all the work you're doing as our country continues to struggle with COVID-19 and the terrible virus that has caused so much death and destruction throughout our country. I want to thank you as well for your commitment to the research that will lead to vaccines and therapies as we continue to confront this challenge. Thirdly, I want to thank you for helping people like me in government, whether members of Congress in the Senate or the House, or members of the administration, helping us to better understand what we're up against as we begin to develop new policy and as we have developed policies these last number of months. All of us in government must listen to you, to listen to the scientists, to listen to the experts who understand what we're up against, and to let science, all of us must let science and public health be our guide. The public health guidance and the science must drive our decisions in government. And we know that's true at the federal level, at the state level, and at the local and county level. So we're grateful for that, and thank you for your continuing work. Also wanted to say how much I appreciate the fact that you've given us good guidance on donating plasma, as I've begun to do after I had a very limited exposure to COVID-19, which resulted in a fever, which was no fun, but certainly I didn't suffer the way a lot of Americans suffered. I was fortunate. And I wanted to try to give something back, and so I've had 2 visits to a local plasma donation facility just near my hometown of Scranton, and I'm getting ready for my 3rd. So if I have antibodies that can benefit others by a plasma donation, that's what I'm going to continue to do. But thank you for the work that you're doing in that area as well. And thank you again for your continuing work on behalf of the American people. I have no doubt that your work, your scholarship, your commitment to excellence, and the great research that you've done already and will continue to do is going to help our country through this great challenge. Thank you, and God bless your work.
Now I'd like to introduce today's speakers. Dr. Mark Dennison is the Edward Claiborne Stallman Professor of Pediatrics, Professor of Pathology, Microbiology, and Immunology, and Director of the Division of Pediatric Infectious Diseases at Vanderbilt University School of Medicine. Dr. Dennison is an internationally known authority on coronavirus biology. For over 25 years, he's conducted NIH-funded research focused on the replication, pathogenesis, and evolution of coronaviruses, including SARS, MERS, and now SARS-CoV-2, which causes COVID-19. Dr. Dennison helped develop Remdesivir, a promising antiviral drug against COVID-19. Dr. Dennison and his colleagues are also studying a new compound, EEID 2801, which has shown promise in mice in reducing lung damage and weight loss associated with SARS-CoV-2 infection. Dr. Dennison is an elected member of the American Academy of Microbiology, the honorific leadership arm of ASM. Dr. Arturo Casadevall is the chair, Molecular Biology and Immunology, and the chair and Bloomberg Distinguished Professor at the Johns Hopkins Bloomberg School of Public Health. Dr. Casadevall is a renowned microbiologist and immunologist who studies how microbes cause disease and how hosts protect themselves against microbes. He's made major contributions to our understanding of fungal pathogenesis and how fungi evade the host immune response. He's developed novel therapeutic strategies for a variety of human diseases, including melanoma, tuberculosis, and now COVID-19. Over the course of the current pandemic, Dr. Casadevall and his team have spearheaded the use of a convalescent plasma therapy as a potential treatment for COVID-19. He has led a team of physicians and scientists from across the United States to establish a network of hospitals and blood banks in several states that collect, isolate, and process blood plasma from COVID-19 survivors. He's chair of the governors of the American Academy of Microbiology, ASM's honorific branch. I will now turn it over to Drs. Dennison and Casadevall.
Thank you very much. It's a great privilege to be here, and I'm honored to be able to speak in this body. I have to say I'm nervous because after so many years, in fact, actually now over 34 years, I think, of working on coronaviruses, I have a lot I'd like to say. I'm going to take you a little bit into the science, force you all to tolerate just a little bit of science, because I think we can't think of this as separate from that. Also, when people ask me what I'm doing nowadays, what we're doing, you must be working on this, they ask me what I'm doing about this virus, and I say, I'm working on the next one. What I mean when I say that is, I mean, we must continue to push forward. We have to be thinking about this in a broader scheme. while we're trying to attack it. And the work that we've done and the work that I'm going to present a little bit of today is really to give you a sense that this is our long-term processes of public-private partnerships, coronavirus basic biology for over 30 years, drug development against SARS and MERS and other bat coronaviruses for the last 7 years. And so Remdesivir and EIDD 1931 did not just come out of the woodwork. Companies, academia had been developing them. We did preclinical development on those, and it's taken many years to do that. And so I think it just points out the importance of contributions and really the commitment to support research in the fundamental biology of these viruses, as well as in potential opportunities for countermeasures. So I don't think I need to tell anybody, which is unusual, that this is a really unique event in human history, I would say. There is no precedent, even the 1918 flu pandemic, not because necessarily of the number of deaths, which is horrible so far, but doesn't match that of 1918 flu, but because this is touching everybody in the world at the same time. It's touching us as an organism, not just in our biology, but in our sociology, our politics, our economies, and the kind of devastation that can be wreaked by such a small, seemingly tiny or invisible organism is profound. This slide also represents and demonstrates that there has been an acceleration of emergence of zoonotic coronaviruses with pandemic potential. There were 4 known human diseases, actually 2 only discovered after the SARS epidemic, but really 4 before that, and probably all within the last 500 years, which from an evolutionary perspective is very fast. And 3 in the last 20 years, all of which have pandemic potential, one of which is manifesting it in really terrible and dramatic ways. So I think what we could say is what's our probability that we're not going to see any more of these? It's low. The probability is high that we will because of human involvement and intervention in human biology or in biology of other organisms. This just shows very briefly sort of an older schematic of an outline of different coronavirus clusters. And in blue are those that are called the beta coronaviruses, from which SARS-CoV and now COVID-19 came. But BT on this slide means bat, it implies bat viruses. There's over 1,200 bat species, 25% of mammalian species are bats. Every bat that's been looked at is shedding at least 2 coronaviruses asymptomatically. And many of those have been identified as what we call outbreak ready. That is, they can infect human cells. So for the last 20 years, me and my colleagues, particularly my colleagues at UNC, Ralph Barrick and his lab, have been predicting and have been proposing that we need to be prepared. We need to be looking for antivirals, not just against SARS and MERS or human coronaviruses, but against future ones. And unfortunately, the future joined us much faster than we thought. So this has really been very profoundly impactful and emotional for us because we have been trying to prepare and now we're being asked to respond. And I want to tell you how we're going about doing that. So this is from our grant application, but this was just the last statement from our 2018 grant application, which basically summarized by saying, with increasing overlap of human and wild animal ecologies, the potential for future severe zoonotic coronavirus pandemic is high. That was our justification for our grant, which proposed 3 things. 1, to look at antiviral drugs, particularly Remdesivir and EIDD 1931, to look at the mechanism of action, to look at the impact on in animal diseases, and finally to look at mechanisms by which we could look at future viruses and try to be prepared for those that might come along. So how can you think about antivirals? Well, I'd like to put it in a bigger context of countermeasures. And by that, that sounds sort of more of like a military application, but it's really not. It means how do we respond? And what I'm talking to you today about really is direct-acting antivirals, those that actually target the virus to try to inhibit its ability to grow inside a cell. And those could be used for treatment. They could be used for prophylaxis and transmission. One of the things we do wrong sometimes, I think, is we think of a drug as, does it work or does it not work? Is it failed or did it succeed? Did it help or did it not help? Why didn't it help? What we need to do is begin to think about compounds more in a sense of how do they work, where do they work, when do they work, in whom do they work, by what route of administration do they work? And then what we do is we stop putting absolutes on things and we begin to think that we have to use these in combinations and cooperatively. We have to encourage development of multiple strategies that could be used to prevent disease, that could be used to treat disease, that could be used to block transmission, that could be used to lessen severity of disease and help people survive. These could be direct-acting antivirals, as I'm going to describe, monoclonal antibodies, which are essentially passive immunization with antibodies that are synthetically made that are known to really act against the virus. And then host-directed therapies, those that would act to block the actual immune response that can be so damaging. It can be so detrimental. And I think many of you may have seen sort of the preliminary news reports about dexamethasone working in that regard. One of our proposals in our grant was to actually look at host immunomodulators in combination with antivirals to try to extend the therapeutic window and just both to eliminate the virus and potentially prevent or treat the complications of it. Excuse me. Okay, so what's our need for antivirals? Well, I think there's no ability of current coronavirus antibodies to stop future coronaviruses that we're aware of. Maybe there's some cross-reactivity, but it really doesn't look significant, and it doesn't look like it's really impacting this virus very much at this time. And as we're seeing now, also vaccine development, even the fastest in human history, lags the trajectory of a pandemic. And next, the potential for universal off-the-shelf use in current and future coronaviruses. So if we can target something that's highly conserved in every virus, and we know it works against every coronavirus past and present, we can make a strong predictions that that might be available to use immediately. And I think that's sort of the case with Remdesivir. The reason it got put in active use in China and now in the United States so quickly is because there was such a profound amount of data that it worked against every coronavirus that we could test. That's important, and that's how we have to think about this going forward. So the idea is you can do multiple things: prevent infections, treat infections, and save lives. Okay. So what are our goals? Well, these are not pharmaceutical goals, except for potentially the last ones. These are really biological, and I'll call them goals of development for us. And those were to look at things that were broadly active against every coronavirus that we could come up with. We're interested in trying to see, can we work with people to develop drugs that have different routes of administration? So people say, IV, that's bad. No, that's great if you're hospitalized and you're really ill. But we'd also like a drug that could be given orally to people who aren't in the hospital, and IV to people who are very ill, potentially inhalational so it could reach the lungs more quickly. So it's not one size fits all. It's not one drug is a cure. That's not a term we should be applying to this virus. We should be talking about how we prevent, how we limit, how we treat. Those are the terms we should use. Finally, we want a high barrier to resistance. We don't want a drug that we're going to use for a week and a half, and suddenly people are going to have resistance and they can't get better because now the drug doesn't work. So that's an interesting goal, and it's one that we've been pursuing. So now a little bit of the biology. Okay, so the coronavirus life cycle. But coronaviruses, like all other RNA viruses, all other viruses, are obligate intracellular pathogens. They cannot live outside the host cell. They can't live on surfaces. They can't grow on surfaces. They can survive, but they're inanimate in that regard. They have to be in a host. They have to be in a cell. So everything you do is you're trying to get something that either stops it from getting into the cell or eliminates it once it's in the cell. I think of it sort of like a factory. You have a parking space that this thing would come into. You have a foyer in the factory. You want to keep it out of the foyer if you can, but if it gets in the foyer, you want to keep it there. And you definitely want to keep it off the factory floor, because this thing is going to rearrange your factory and create new factories to make more of itself. So there's multiple places you can stop that. So it has to get in, it has to take over, and then it's going to damage the host cell. And then what happens is the host is going to attack that cell because it's virus infected. And this is where some of the dramatic inflammation can occur, which can be as or more detrimental than the virus infection itself. So what are the stages? I'm not going to go through these in detail, but I just want you to understand there's a simple life cycle, and this has a lot to do with how we think about targeting the virus. The virus has to attach and enter the host. Okay, it enters by receptor binding on what's called ACE2. Once it gets in the cell by one or more mechanisms, it opens up its RNA genome in the cell, and that is translated into proteins. Those proteins then do work to rearrange the whole it rearranges all the furniture, all the membranes, all the stuff in the cell to make its own virus factories. And then those proteins go to work copying the virus genetic material, which then assembles into new virus particles, which are then released to infect other people or other animals. In the case of this virus, it can do both. So these are all steps by which you can potentially inhibit virus replication. So if we're thinking about attachment and entry, this is an area where you may have heard about monoclonal antibodies that can bind onto the surface of that pink spike protein, looks more like a mushroom than a spike, my wife informed me, and that can block it. It coats it so that it can't see the receptor and can't get in the cell. This is also where plasma would work theoretically, because you would be having antibodies that would be capable of stopping the virus from getting into the cell, getting into the foyer, getting onto the factory floor. If the virus is taken up into what's called an endosome, which is an internal vesicle, another place you can stop the virus is by doing something that prevents that endosome from fusing with the virus. This is where chloroquine, if it has an activity, is proposed to work, by changing the environment of the endosome so the virus can't release its genetic contents into the cell. This is also a place where natural antibodies would work, where vaccines would work by preventing the virus from getting in the cell in the first place, and potentially ACE2 inhibitors that would block the receptor of the virus. Okay. So after that then would be these issues of the virus translating its proteins. Coronaviruses make one big protein, call it a polyprotein. It's like a chain of pearls on a string. And proteases are with the virus scissors that cut those proteins apart. And so proteases are very important. Other viruses use them, like HIV. So protease inhibitors, and one that's been proposed is lopinavir-ritonavir, which is an HIV protease inhibitor, that would block this process. And there are others that have been tested in some coronaviruses, but not in human coronaviruses. So this is a potentially very powerful place to block the virus. And what I'm going to talk about for the few minutes remaining is really remdesivir and EIDD-1931. These target the part of the replication that I consider the key, because of course I've studied it my whole life and I think it's the most important, which is the place where the virus sets up its factories and starts to copy its genetic material using enzymes called polymerases. They copy and they copy the genome so that it can be put into new viruses. So these drugs act specifically to inhibit or act at the level of the polymerase to screw up viral RNA synthesis. So they can be profoundly active. They're used in these kinds of drugs, nucleoside analogs, are used in a wide variety of viruses, like acyclovir for herpes viruses, or sofosbuvir for hepatitis C, or AZT for HIV. These are nucleoside analogs that block virus replication. Finally, I just want to mention before I come back to those drugs, the issue of how can we prevent or block cell damage and inflammation. And this is where dexamethasone, if it works, would work, where IL-6 inhibitors wouldn't work, by blocking the steps or pathways or endpoints of inflammation. You can't do that by itself. They've been detrimental when they've been used with SARS or other things, because each virus is independent, and if you block the immune response too soon or at the wrong time, you can allow the virus free rein in the body. On the other hand, if you use it at the right time, and you just use it to limit damage that the virus causes while treating the virus or while the virus is being treated by the body, then you potentially can help people recover from infection. So as you can see already, combinations of drugs is really the mechanism by which we need to be thinking about this and other coronaviruses. So I will just tell you now some very summary slides, which just hurts me to just give you summaries, but I'm going to have to do it. And this is what I call the polymerase complex. This is what I've been working on for my entire career. The viral polymerase and all the proteins that go along with it, the unique functions that coronaviruses have to evade lots of different things in the body and the cell, then keep going. So this is more than 30 years of NIH-supported basic science in model coronaviruses, one called mouse hepatitis virus, as well as SARS and MERS since 2003 and 2004, and over 7 years of drug development. So I think what you can see is that if we have anything that works that comes out of this, it's been because there's been a long-term commitment. And this is just the two drugs that I'm going to mention briefly. Remdesivir, which you may have heard about, which has moved forward pretty aggressively and has made it through the initial studies and now is being used as part of the ACT trial. And then EIDD-1931. Both of these are nucleoside analogs. And what these graphs show in a very simple schematic way is that if you increase the dose, you decrease the amount of virus. And those numbers on the left are logarithms. So what that means is virus titer reduction of about 1,000,000-fold at low concentrations of these drugs in culture. So these are very effective drugs. They work by different mechanisms, but they work and they're independent. And so the exciting part is that they might represent unique and independent ways to inhibit the virus. So just to summarize, remdesivir potently inhibits multiple divergent coronaviruses: mouse hepatitis virus, SARS, MERS, bat coronaviruses, human coronaviruses, and now SARS-CoV-2. It has a high barrier to resistance, meaning we tried to get resistance to this drug in the culture, and we had a very difficult time. And the virus that became resistant was much weaker and damaged and couldn't compete with the natural virus. So resistance exacted a cost. It can prevent disease in animals, both in small animals and in primates. It can decrease lung disease in primates. It's active against SARS-CoV-2, and that's been demonstrated now in people as well as in the lab. And its safety in humans has been well demonstrated in more extensive trials in Ebola and now in humans with SARS-CoV-2. So this is a very exciting opportunity for this drug to move forward really remarkably quickly for a drug in development for a new mechanism. I want you to see that this, again, maybe I'm making my succo too impactful, but it's important. What you can see is our first interactions with Gilead. This is a public-private partnership between Vanderbilt University Medical Center, Gilead Sciences, University of North Carolina, Chapel Hill, University of Alabama, Birmingham, and others. And that's been NIH supported now since really 2014. When we first contacted Gilead and got the compound to test and saw its activity, followed by studies in my lab and that of Ralph Barrick that went on to study this in animals and in the lab with publications in 2018. Ongoing support showing that this had a broader spectrum than we thought, that it was superior to lopinavir ritonavir against MERS, and that it was just ready when this pandemic began to occur and could quickly be implemented. But you can see that only occurred because of that preparation that we had been doing and the planning and our doggedness about the fact that this was something we were likely to see in our lifetimes. So I'm just to summarize a couple more points. One is about EIDD 2801. This is another compound developed out of Emory University, Emory Institute for Drug Development, and that is a compound. And the difference in this one and remdesivir is an IV drug, so it's been used in the hospital for ill and severely ill patients. EIDD 2801 was less far along in its development, but it has shown it's met all the same milestones, as you can see by the green check marks here. And now there has been safety testing in humans and safety and tolerability testing once this was licensed by Ridgeback Biotherapeutics and now licensed by Merck for further clinical testing and development. We're excited about this compound because it meets all the similar milestones as remdesivir, and it has an oral bioavailability, suggesting the opportunity for using it as an outpatient or using it potentially in other combinations. It's not there yet. It needs to be thoroughly tested for safety and for other factors that are associated with its chemistry. But nevertheless, it seems to be something that can move forward into that scope of testing. So what are we doing? What are our final goals? One is we're attacking SARS-CoV-2, but we're preparing for what I call SARS-CoV-3. Heaven help us. We cannot stop thinking about coronaviruses and their potential because of what we've seen in bats, what we've seen in humans, and what we've seen about the scope of this. There are 7.3 billion or more immune-naive, susceptible people to this virus. This virus is not under evolutionary selective pressure from us. It has a long way to go, and we need to be very aggressive at continuing to look for mechanisms to treat this and prevent it. We need to look for new targets, and we have many good ideas I'd love to talk to anybody who's interested about. We need to look at combinations of immunomodulators and direct-acting therapies. We need to look at using monoclonal antibodies in plasma. Okay. We need to look for multiple routes of administration. So finally, my closing comments are that these two drugs are available for human testing because of greater than 7 years of committed work and support for coronavirus basic science and drug development from the NIH, from the CDC. Some has come from DARPA, some has come from DITRA, some has come from other organizations, and there has definitely been a partnership with Gilead and with others for this support. Support for countermeasures has to be a long-term commitment, and we can't abandon one virus when another one comes along, because we don't know the next family that's going to happen, although I would predict another coronavirus would be a likelihood. So finally, it's very challenging for labs to do this work. It's very challenging for young investigators to do this work. And so not only do we need to support the kinds of studies to find the targets and develop them, but we need to support the people who are training in it so that they're incentivized to be sure that they want to go into this kind of field.
Thank you for inviting me. I want to be talking about a different approach from the one mentioned by Dr. Dennison, although I believe that they're complementary. This is the convalescent plasma therapy for COVID-19. So the most important, so what is convalescent plasma? Convalescent plasma is the plasma, the liquid part of the blood after a person recovers from COVID-19. As you heard Senator Casey, it is obtained from people who recover from the coronavirus. And it's also important that he noted that he's given it twice and he can give it 3 times. So the other important thing is that every time that he donates plasma, he can treat 2 to 3 people. So the mathematics here, the arithmetic is in the favor of this. People who survive can help those who come afterwards. So what is the active agent? The active agent is antibody that neutralizes the virus. It differs from a vaccine, that when you get a vaccine, you make your own antibodies. When you get a plasma transfusion, you get in the antibodies that other people made. And so this is straightforward and very simple. A person recovers, they have antibodies in their blood. When they're well, they donate their plasma, which is the liquid part of the blood, and then this is used to treat people or to give it to them to prevent a disease. So this is old. The first Nobel Prize in medicine was given to Emil von Behring for the use of serum therapy. This was state of the art on the turn of the 20th century. And it was the first time that physicians could treat anyone with an infectious disease. And you see there a picture from Scientific American in 1894, a child being treated with serum, and the success of serum against diphtheria, a major killer of children, was so striking that the first Nobel Prize went for this method. It's been used in many past epidemics. In 1918, it's been used in local outbreaks, and I'll tell you more about that. For example, in a school to control measles, and as late as the 1940s, it was used for the treatment of mumps. So here's a summary. It was used in the 1918 flu. It's been used to control outbreaks of polio, mumps, and measles in the United States. In the 1970s, it was used to treat Argentine hemorrhagic fever. SARS-1, coronavirus in Hong Kong, was treated with plasma. In 2012, the H1N1 influenza epidemic. There have been some anecdotal reports of the use of plasma for Middle Eastern respiratory syndrome. For the Ebola outbreak in West Africa, plasma was successfully used. And now I'm going to tell you about the use of COVID-19. So before we get there, though, history gives us a lot of lessons, and they are very important for going forward. Antibody is effective in the treatment of infectious diseases. Efficacy depends on timing. The earlier, the better. If you're going to use plasma, you want to use it when people begin to get sick. It doesn't work as well when people are intubated in intensive care units. The efficacy depends on the amount, and a certain amount is needed. You need to have antibody in there, and the more antibody you have, often the better you do. So antibody therapies were well tolerated, and when people recovered, the recovery was rapid. Pneumococcal pneumonia, which is also known as walking pneumonia, was treated with serum before there was penicillin. And when penicillin came, they made the observation that when people got serum, they felt better rapidly. Well, it took a few days to get better with penicillin. And the reason for that is because there's a lot of stuff in plasma that can neutralize a lot of the things that make you ill. But these were largely abandoned in the 1940s. And here you have to think about bacterial diseases. For bacterial diseases, they develop antibiotics. And so plasma and serum therapies could not compete. By the way, you'll hear me say plasma and serum interchangeably. And that is because the historical term is serum. What we talk about it today is plasma. It just means that different preparations of the liquid. But in reality, for what you need to know, you can hear them and think about them as interchangeably. For viral diseases, the problem was that they didn't know about hepatitis or bloodborne pathogens. And consequently, there were some outbreaks of hepatitis, and then this put largely an end to this approach. So Wuhan 2019, I happen to be very interested in history, and I know a lot of this history. By January, I was very concerned that this was not containable. I also knew that the history of antibody therapies was not well known. I kept listening to our public health officials and did not hear the word of plasma therapy. And I wanted to try to get the word out. And one of the questions is, how do you do it? Well, I could have written an op-ed or a letter in a medical journal, but that wasn't going to work. You really needed to put it in a public place. And I decided to try to write an op-ed and began looking for a paper. Unfortunately, the Wall Street Journal took it, and this was how a boy's blood stopped the outbreak. And this is something from that paper. Basically, you had a school in Potsdam, Pennsylvania, known as the Hill School, and the doctor, or the school doctor, noted that a kid had come down with measles, and he had 66 kids in the school that had not had measles. So what he did is he waited for the kid to recover, and then took the blood from that kid, spun it down to get the liquid, and then as little as 10 milliliters was given to the other boys. And the result was that he stopped the outbreak. So I used this to frame the op-ed. And then you can see the op-ed was published in the Wall Street Journal in February 27th, and then I sent it to all my friends. Who are all my friends? All my colleagues, anybody I could email it to. And began to have discussions at Hopkins, and we assembled a team at Hopkins to try to deal with this. This was in those early days of March when things were starting to spiral out of control, and it wasn't quite clear. But I realized that we needed to have a manifesto, something that had all the references that could be given to physicians who were going to write something for their IRBs or regulatory agencies. So we wrote a paper with Lisanne Porofsky, and the Journal of Clinical Investigation had it reviewed in about 3 days and published right away. And then all our friends, we assembled to create the National Convalescent Plasma Project. Key people here are Michael Joyner, Mayo, and Nigel Panek at Michigan State University. Others are Jeff Henderson, Lisanne Porofsky, Shmuel Shoham. We all came together as a collection of physicians, scientists, wanting to do something about this. And within a few weeks, by March 24th, the FDA allowed compassionate use, and March 27th, the first patients were treated. This is what I call COVID time. This, what you see in that slide, would have probably taken years. But in the emergency that we were dealing with, it was possible to move very fast. And I hope one of the legacies of this time is that when we look back, that we learn to do things a lot faster than we were doing. This is our website. By the way, industry stepped up to help us. The website was set up by Amazon. And so imagine this as a grassroots effort of citizens trying to do something about it. They happen to be physicians and scientists, and we're getting help from private corporations. So, and the FDA moved very rapidly, and I want to commend our FDA colleagues who have really been partners in this, in particular Peter Marks and Nicole Verdon, and the Mayo Clinic team, Michael Joyner and Scott Wright. So you see the dates there, March 24th, April 3rd, April 8th. Within a couple of weeks, the FDA had allowed the use of plasma for hospitalized patients, in particular if the doctors made a determination that it could be useful. By mid-April, this is the number of sites. It was about 1,000 sites in the United States that registered for the use of plasma. Today, the number is over 2,500. The two publications that we did, the first one was what I call the manifesto, which was just putting out the information on the option. This reviewed the history, and it brought people up to date, to speed, basically, on what could be done. But then we realized that we needed a how-to guide, because in reality, nobody was giving convalescent plasma in the United States, and we needed to have like a manual. What do you need to do? What things to check? And this was written by Evan Block and Aaron Tobian. These are all the collaborators that contributed. And these are transfusion medicine people. One of the fascinating things about this is that this group includes transfusion medicine, infectious disease, epidemiologists, immunologists, virologists. It's a very much interdisciplinary group. Both papers are available for downloading. Then China began to describe the use of convalescent plasma, and 3 papers appeared in late March, early April. And even though these were not what you call controlled clinical trials, they're case series. What it means is that they're describing the use in patients. And even though they are not that good for giving you a sense of efficacy, they were very encouraging. These people were being treated with plasma, and the plasma was working as an antiviral agent. They were basically suppressing the virus, and they were reporting good results. And the other important things about case series is that one of the great concerns at the beginning of this was, can you hurt people by giving them this antibody? There had been this literature of antibody-dependent enhancement, whereas if you give an antibody, could you potentially enhance the virus? And this provided some reassurance that this was not going to happen. Italy began using plasma too, and even though It was initially at the newspapers. I think you can make out some of the La terapia con plasma funciona, which from my Spanish, I can tell you the therapy with plasma works. And this has been published as a preprint recently, and here is the preprint. Sorry you can't read it, but just know that the information is now out and available. So the situation today in the United States, about 20,000 patients have been treated. National and international networks are emerging. Convalescent plasma has been used throughout the world in different ways. Some nations have moved very rapidly, others have not. But I can tell you that the United States is probably the leader in plasma, and that is a credit to the response of our physicians, the response of our institutions, and the response of the FDA, which moves very rapidly to allow this to be used. Convalescent plasma supplies were initially very tight because you needed to have donors. But today, convalescent plasma is more available, and in many places in the United States, people who are sick have access to it. Convalescent plasma is also being used to produce a hyperimmune globulin, and there are numerous clinical protocols ongoing, including randomized clinical trials. But the big questions everybody want to know is it safe and does it work? So let's take safety first. Oops, I keep hitting. Here we go. Safety. So we published the first paper in April on the first 5,000 individuals. And I can tell you that the safety profile is the safety profile of plasma. All the concerns about antibody-dependent enhancement, triggering cytokine storms, none of that came to be. And at 12 o'clock today, we published the experience on 20,000 patients, which is available from the Mayo Clinic proceedings. And the data, again, the safety profile is very good. We don't have any indicators that giving specific antibody to these patients is doing any harm. Is it working? Well, there are hopeful signs. In fact, everywhere you look, you find hopeful signs. You have several case series from China were encouraging. I mentioned that. Italy reported a 50% mortality. Mount Sinai in New York reported a 50% reduction in mortality if used before people went into the ICU. This is available as a preprint and it's in review. And here is the mortality of people before they went into the ICU. Again, the theme, it needs to be given early. Once it's given to individuals, later it is not as effective. The first randomized controlled trials, one of them was beginning in China, but there was no conclusion on mortality because they had to stop the trial before they ran out of patients, essentially. That paper was published last week, and additional data is expected very soon. This is very interesting data that I'd like to share with you. It was just available as of a couple hours ago, and it's in the Mayo Clinic paper. Look at what happens. This is the mortality on the 20,000 patients. And look how it's dropped from about 15% down to about 5% here in the United States. Look, we don't know what the explanation of this is. It's good news. You want certainly your seriously ill people to be dropping, but all these patients receive plasma. And one of our interpretations of this is that physicians are learning how to use it and are learning that they have to give it early. So whereas here, in the early weeks of April, they were using it as salvage therapy, they were only giving it to people who were really, really sick. Today, they're probably using it a lot earlier, and we think that that is an indicator, suggestive of a reduction in that the plasma is working and is reducing mortality. So my opinion on efficacy, and I stress in my opinion, there is strong precedence for efficacy based on historical use. There is a strong theoretical support from efficacy based on what we know about antibody action. We have encouraging signs from case stories with matched controls from the United States, Italy, and Sub-Saharan Africa, 2 countries, Burkina Faso and Senegal. There is one randomized from China, that was prematurely stopped, also provided encouragement. So people ask me, is it working? Well, I give you what is my efficacy descriptor. If you asked me in March, I would have said to you, maybe. If you asked me in April, May, I would have said to you, possibly. If you ask me today, I would probably say to you, probably. But I will not say definitively, because definitively requires a much higher level of rigor and knowledge that we need to go do randomized clinical trials. And what we hope to be able to say definitively in the future. So here at Hopkins, we have two trials that to try to prove that work, and these are outside of the hospital. Hopkins is focused on the outpatient space. So one of the trials is a prophylaxis. That is, if you have individuals with high exposure, potentially in a nursing home, first responders, if you gave them a unit of plasma, do you prevent disease? And the other trial is an ambulatory protocol, which is looking at people who are sick at home. And if you give it a unit of plasma, do you prevent them from getting worse? Both trials are ongoing. These are the individuals leading them. Shmuel Shoham, Dan Hanley is involved in both trials, and David Sullivan is involved on the ambulatory protocol. These are multi-site trials. We have obtained support from the Department of Defense, who's very interested in this, because clearly, if you can use plasma to prevent disease, it has tremendous implications for military readiness. So the near and far horizons. Today, convalescent plasma is available in the United States, in most hospitals in the United States, so doctors can get it for you. There's a lot of people donating. It's being donated to the Red Cross. Hospitals are collecting it. So, and the efficacy is, I won't say definitively, but the indicators are that it's been effective. In late summer and fall, we are hoping for immune globulin, which is derived from plasma. But these plasma studies will inform the use of immune globulin. Immune globulin just means that they take the antibody out of plasma and they make a pharmaceutical preparation that's standardized. So it's a better product for using clinically. And hopefully that will be available, but it's going to take some time to get here. Monoclonal antibodies have been developed by many groups. And again, convalescent plasma will inform on the use of immune globulin. For example, one of the things that we are learning with convalescent plasma is how much do you need to get efficacy? Well, you can take that information and then use it for immune globulin and monoclonal antibodies. And convalescent plasma is also going to help vaccine efforts. I give you an example. Those two trials that you help at Hopkins, if we show that convalescent plasma can protect against disease, you could rapidly hasten the development of vaccine by using challenge studies. What that means is that you take volunteers and then infect them, because if they get into disease and into trouble, you could then treat them with plasma. You have something effective that can be used. And the safety of convalescent plasma will accelerate vaccine studies. One of the concerns in the coronavirus field was whether, in fact, a vaccine would work, because there were these concerns about antibody-mediated enhancement. But now establishing that human antibodies are safe in humans really lifts a lot of those concerns away. So I think that is my last slide, and I thank you for your attention.
Well, thank you so much, Dr. Dennison and Dr. Casadevall. Now we're going to move into our Q&A portion for the last few minutes we have. So please do submit questions if you have some that have come up during the presentations. I'm going to start with Dr. Dennison. You mentioned the course of the many years you have been looking at antivirals, in particular Remdesivir, but can you talk a little bit about why does it take so long to really prove they're effective, and what, as you're looking at a potential candidate, what are really the most essential things that you need to know in order to move the research forward and get to an effective therapy?
Well, I think I would hope that we could accelerate, as Dr. Casadevall said, that this is a new era and that we can accelerate it. There was no time, unfortunately, to cover all the different aspects, but I think one thing that's happening right now is sort of really high throughput rapid screening processes. that allow us to quickly determine if something has potential or not. One really important one is called repurposing, where there's drugs that may have been used against another virus or may have been used for an independent purpose and are known to be safe in humans. And so a lot of the issues in timing and length of time is about making sure the pharmacology is good, making sure they're safe, making sure they don't cause any harm. And so I think we could accelerate that. Ours was a longitudinal long-term study because it was brand new. It was new in coronaviruses. The work only supported longitudinal studies, and it was fairly directed. And this was a brand new process that had not been done. In my opinion, I think there may have been 3 or 4 labs in the world, certainly only 2 in this country, that were highly focused on coronavirus antivirals, mine and along with Outbreak being one of those teams. So I think part of that was just the nature of how much support there was for that process when there wasn't an active disease like SARS and MERS was sort of away in the Middle East. But I think that the opportunity to really accelerate that is there if people pay attention. You still have to go through the process of testing in vitro. You have to make sure it works against the virus, against multiple viruses. You have to make sure it doesn't kill cells or cause toxicity. You have to make sure it doesn't cause adverse other events to the cell. You have to make sure it would work in animals and or in humans with good pharmacology, not interacting with other drugs, not causing problems. So there's a lot of things that have to be done. But I think there's ways that those can be done now in parallel, like is being done with some vaccine studies. And I think that the kind of approach that Dr. Casadevall is describing for plasma is something that could accelerate a lot of drug testing.
Thank you. Dr. Casadevall, did you want to add anything based on your recent research and looking at the course of history working with convalescent?
So I think that to beat this disease, we're going to need to have layered defenses. We're going to need a lot of things. It isn't going to be antibodies. It isn't going to be a vaccine. We're going to need drugs, antibodies, vaccines. And the other important thing is that the history of antibodies is that they work very well with drugs. So I would look forward to see combination therapies in which you get both either plasma or gamma globulins or so, Remdesivir and some of the other antivirals. And I think we can really bring down, if we can bring the mortality of this disease way down, you'll take the terror out of the pandemic. And if you do that, you will really be able to reopen society.
So this question came in, and it's really for either or both of you. So how can Congress continue to effectively help scientists like you expedite the research, the development, the manufacture, and really the distribution of these treatments? Just from your vantage point, what can be done, and what is being done, and what can be done?
Arturo, I'll let you go first.
Well, I just want to say that both Mark and I have been supported by the NIH fund, and the biomedical defense establishment is the first line of defense against this. Aircraft carriers don't help you. F-16s don't help you. And this is a biomedical research establishment that has been hurting and underfunded for the last 20 years. Mm-hmm. And yet it's risen very rapidly to the challenge. And it's now providing options, almost on a weekly basis. So what I would say is that what is needed is to think about this as a human resource that can be deployed in an emergency. And this is what is done. And Congress, I would hope, would work to stabilize funding for science, and particularly basic science, because the fundamental work is ultimately what drives everything. And so not focusing on things that are only useful, focusing on basic science, focusing on training people, and providing stability so that the scientists are better prepared when COVID-3 shows up.
I'd only add that, as I mentioned, the basic science, what I'll tell you is that the reason that we reached out to Gilead in the first place was because our lab had discovered this function in coronaviruses called proofreading, which doesn't exist in any other RNA virus. It's unique, and it prevented them from being inhibited by many nucleoside analogs. And we wanted to know, is it possible to find one that would work against this unique function? This was a paper that had very few citations and very little visibility, published in Journal of Virology in 2007, but now has been identified as a key novel target for coronavirus antivirals, and people from many different fields that have exonuclease inhibitors have contacted us. So I guess one of my points here is that that would not have been something that people would have thought if someone had looked at that in Congress. Why are you studying an exonuclease of a mouse virus, a mouse coronavirus? What has that got to do with human health? And what we know now is that is the key linchpin, keystone enzyme that protects coronaviruses from many kinds of different drugs. And so that is something we started working on in 2003 or 2004, long before this happened, right after SARS. So this just points out to me, I think what Arturo was saying, is that we have to stay focused on doing the basic science, understanding the targets. We have to understand not just screening for drugs, but we have to have targeted approaches that are really rational as well. One other point I make is that we cannot abandon one virus when another one comes along. We can't abandon Zika when Ebola comes along. We can't abandon Ebola when the coronavirus comes along. We can't abandon paramyxoviruses when there's another flu virus. Any of them could do this. Some of them have more potential to be more severe, but no one before 2003 had coronaviruses anywhere on their list of viruses that might cause severe human disease and pandemics. Viruses are smarter than we are, I'm afraid. And we have to be broadly prepared because we cannot predict where the next threat is coming from.
Well, we are at 3 o'clock, and I think that's actually a perfect way to end today's webinar. I want to thank you both again so much for your time and your expertise and for your outstanding presentations. And I want to thank all of you for joining us today. We also have made our 2 previous programs in this series, one on diagnostics and one on vaccines, available at www.asm.org/advocacy. And with that, I will conclude our program today. Thank you so much.